we're going to talk about the role of excipients in making of a robust formulation. This is the typical formulation practice that we all adopt, which starts from the literature survey. Then we have the selection of excipients, the trial batches, testing of formulations, the pilot studies, exhibit batches, stability, bioequivalence, ANDA, review, and finally the approval. This is the conventional approach that we all have been following. And this has certain disadvantages, like it's poor understanding of input and response. There's no relation between input and the response. Non-conformities are there. Then you have to investigate the non-conformities, rejection of batches, the manufacturing cost because of this goes up, and we have the change in process made very difficult. So, is there any alternative approach? Yes, the approach is the QBD approach. So, we'll see how this approach will help us in selecting the excipients. So, this is the concept of QBD, which essentially puts the thrust on planning the things at the times of developing. That means the onus of quality is shifting from QC, QA to the R&D. Where you are developing a formulation, you should ensure that you are planning the things in advance. So QBD can be defined as a systematic approach to development that begins with a predefined objectives and emphasis on product and process understanding based on sound science and quality risk management. So first we saw the drug development by the conventional way, and now we see it by the QBD way. So we first identify the QTPP. For example, if you're making a sustained release formulation, so SR is a QTPP. Then we identify the CQAs or the critical quality attributes. In this case, with the release profile. We link it with the critical material attributes and critical process attributes. We perform risk assessment, develop a design space, develop a control strategy, and manage the product throughout the life cycle. So first we saw the conventional, and now this is the QBD approach. Now what this QBD expect is with respect to the excipients. So formulation development by the QBD way with reference to excipients will have the critical quality attribute of the formulation as our objective, and knowledge of various physicochemical and performance characteristics of the excipients will help us to carry out the design by the QBD way. I'll give you one example. Q3D, ICH Q3D is in. So we need to define the elemental impurities depending upon the daily intake. With respect to that, we have to go backwards and find out what are the excipients we are using and how they are going to impact our final formulation in respect of the elemental impurities. So what a formulation contains, essentially, it has three major things: API, process, and excipient that makes the product, and the role of excipient is increasing with the drug doses going down and down. So a formulation is like a movie where API is a hero. The movie is incomplete without the heroines, the excipients. So the excipients play a very vital and major role in the formulation that we'll see in the further presentation. We are going to basics. What is the API? It's a pharmacologically active material which has its effect due to its chemistry. Underline word chemistry. As against that, excipient is an inactive substance which has been appropriately evaluated for safety, which serves as a vehicle, processing aid, etc. of a formulation, and its complex performance evaluation only can assure its efficiency. So the essential difference, in one case, by knowing the chemistry, we can conclude about the quality. In other case, chemistry has hardly any role to play. So the complex role of excipients in a formulation, unlike API, the effect of excipient depends on various physicochemical and other parameters that defines its efficiency, thereby affecting the ultimate therapeutic effect. So the therapeutic effect of the formulation is being affected by excipient in a very complex way. So it's quite easier to establish the quality of API by chemically testing it through a pharmacopoeia. For example, paracetamol, if you are testing paracetamol by USP, you are almost sure that it's going to perform. Against that, if you test, say, magnesium stearate by USP, is passing, you can't say how it's going to perform in a formulation because surface area, which is a critical parameter of magnesium stearate, is not defined in the USP. So establishing the chemical equivalence, which pharmacopoeia normally does, will not help you a lot in determining the quality of a excipient. So these are the various parameters like particle shape, particle size distribution, surface area, flow property, molecular weight, density, viscosity, crystallinity, solubility, moisture, purity, assay, compatibility. And if you see, most of them are physical parameters than the chemical one. So there are many desired grade of excipients because there's one paracetamol, but MCC was 1, then it became 101, 102, 112, 200, and now we have more than 15 MCCs available. So the different manufacturers, different processes, and different analytical methods, because they are sometimes not the pharmacopoeial methods, generate the variables in the design. And we need to take care of these variables if you want to ensure the quality of our formulations. So why QBD is important for the excipient selection, the million-dollar question. If the raw materials are variable, the process is a fixed, validated one, but your end product may not be the fixed product, but it will be a variable product. By this, what I mean to say, for example, you are choosing a magnesium stearate. The surface area range is varying from 5 to 15. First, you are having in the range of 6 to 7 meters square per gram, and next time is 13 to 14 meters square per gram. Your formulation may not behave in the similar way. So going by today's pharmacopoeial testing of an excipient, we cannot assure consistent performance of a formulation throughout the life cycle. So we need to carry out some very important evaluation in terms of excipients to ensure it's going to perform well in the formulation. So the current approach versus the QBD approach. If you go by the current approach, the quality is assured by testing and inspection. Against that, in QBD, it's built into design based on scientific understanding. There, it's based on the data submission, where the QBD is based on knowledge-rich submission. You don't have to generate a lot of data. The process, conventional process, is rigid, whereas QBD is flexible. Focus is on reproducibility. Against that, in QBD, the focus is on the robustness. That's why if you want to develop the robust formulations, we have to go by this QBD approach. And there we are history-based specifications. Against that, we have performance-based specifications. So why excipient testing, performance testing has to be done? A study is presented to highlight this effect. This is a very important study which will tell you what the point we are going to make. And magnesium stearate we have chosen because it's the most extensively used excipient. It is used in more than 90% of solid dosage forms. And there are a lot of different manufacturers making this product. And that's why the surface area plays a very important role. And there are density and particle size distribution are also important, which vary from manufacturer to manufacturer, also vary on the process of the analytical process of measuring the surface area. So this is the marketed preparations with respect to surface area. We found a lot of variation. Now we look at the ideal powder flow properties. One is Carr index or the compressibility index. Other is the Hausner ratio. So if it is less than 10, the Carr's index is perfect. If it is going to touch 38, it's bad. Same way for the Hausner ratio, 1 to 1.1 is excellent. If it touches 1.6, it's not a very good Hausner ratio. So now we share a research paper published in a journal. This research was carried out by Ajanta Pharma, which is one of the top 5 pharma companies in India, and published into IJPSR in 2012. This study emphasizes on the decisive role of particle size of magnesium stearate, its concentration and lubrication time to establish the desired quality attributes of formulation containing the highly soluble drug. So the samples were made using one Nitika Tablube and other brand of MGST. The blending time was 5, 10 and 15 minutes. So if you see the compressibility index, you'll find with the Nitika material, you're getting good results with 5, 10 and 15 minutes. With the other material, the results are only fair with 10 and 15 minutes. As against, if you look at the Hausner ratio, in 5, 10 and 15 minutes, you're getting passable result in 5 minutes, and they are improving as you go to 10 to 15. In case of the other magnesium stearate with Nitika, the results are good and excellent only. So the same formulation, just change the excipient, and you can see the strikingly different result in the Kars index and the Hausner ratio. If you blend for 5 minutes, you'll have a good compressibility, but only passable Hausner ratio. If you go for 15 minutes, your compressibility index will not be so good. That's the first point. We come to the next criteria. There's a friability index. If you look at the 2 bars, one is Nitika Tablube with the other material of magnesium stearate, the dark blue colored bars, and the friability is around 0.3 with the Nitika Tablube. As against that, it is crossing 0.5 with the other magnesium stearate. So same formulation, some change in the excipient like magnesium stearate, and you can see strikingly different results in the friability. The DT or the disintegration time. Again, the concentrations used are 0.75%, 1% and 1.25%, and the blending times again are 5, 10 and 15 minutes. With the Nitika is a cream colored bars and the other material is a dark green colored bars. So if you look at the across the board with the Nitika material, the disintegration time is between 8 and 10. So 8 and 10 minutes, and against that with the other brand of magnesium stearate, it is crossing 18. So the disintegration time has doubled in the formulation because of the change of one critical excipient, the magnesium stearate. And now we come to the most critical attribute of a tablet, that is the dissolution profile. And there are 6 curves here. Out of 6 curves, if you see closely, the 3 curves are superimposed, and the 3 superimposed curves are N5, N10 and N15. This is the 5 minutes, 10 minutes and 15 minute blending time with the Nitika magnesium stearate, whereas the other curves are M5, M10 and M15 with the other magnesium stearate. If you look at this curve closely, you will be able to find out that in 5 minutes and 10 minutes, you have a dissolution of less than 40%. As against that, it is more than 80% with the Nitika material. So this is the very striking result, which tells us we are making the formulation, we are validating the process, but we are just changing a source of one excipient magnesium stearate, and dissolution is totally going double. Also, these 3 curves are not superimposed. So that tells us the blending time has a great impact on the dissolution, whereas with the Nitika material, all the blending times are superimposed. So this also tells us another observation, that blending time has no effect on the formulations with this magnesium stearate. Now we are going for 1% magnesium stearate, and the results are similar. You are having about 35% with the other source, whereas with Nitika, again, it's touching 75 to 80. And with the 1.25% concentration of magnesium stearate, the dissolution with the other brand has further gone down to 30%. Against that, Nitika is about double. So with the different concentrations also, the resolution has been almost double in some cases if the excipient, in this case magnesium stearate, is not appropriately selected. So the conclusion of the study is, as demonstrated in the research article, change in the particle size, hence the surface area of magnesium stearate, along with the concentration and lubrication time, can affect the properties of blend or tablet significantly, such as DT, hardness, and most importantly, the dissolution of the profile of the tablets. If you are having the QBD approach, we'll evaluate the performance of excipients. So we'll have the desired CQA, that is dissolution profile and DT. If we go by conventional approach, our formulation may not have the desired quality attributes. So this is demonstrated by this visual. If you want a coat, you go to a shop, you can have a loose fit or a tight fit. But if you want the tailor-made fit, you have to go with the QBD approach. So the QBD approach will give you the correct fit. So quality cannot be tested into the products. It has to be built in by the design. The logo of Nitika is our final product, the happy customer. Thank you very much for your time. If any questions, thank you. If any questions, we also have a stand in hall number 8. It's 8G121. So if you like to come there, and I mean, it's the time, almost everybody will be leaving, but if you have time, you are most welcome to be at the stand. Yes. Okay. So thank you everybody. Thank you for your time.